Hi everyone, and welcome to today's webcast, Finding Low-Level Residues in Dirty Feed Samples, Developing Robust and Reliable LC and SMS Methods for Testing in Complex Matrices. Today we're joined by Kurt Durand from Eurofins and Syed Sajjadi from Phenomenex. They teamed up to develop a new multi-class residue quantitation method to identify low-level contaminants in feed samples. Our first speaker will be Kurt Durand. He'll give us an overview of the scope of the method and details around optimizing the sample preparation to extract the compounds of interest and reduce the matrix interferences. And then following Kurt, Syed will describe the method development procedure initiated to select the appropriate HPLC column and conditions and to optimize the chromatography to monitor the residues of interest. If you have any questions for Kurt or Syed during the webinar, you can write those questions into the box that's going to show up right now on the top left of your screen. And they'll answer some of those questions at the end of the seminar. Also, you can download an overview of this application note from the box at the bottom of your screen at any time. So now I'm going to turn it over to Kurt to get us started. Kurt?
Thank you, Lauren. As Lauren said, we're going to be covering a little bit on the method development and sample preparation work that we covered here at Eurofins, sort of giving you a background on how we came up with this multi-class, multi-residue method. And then we have some sort of unique analysis using the ABSciX instrumentation that we came up with using polarity switching, which is kind of nifty. So I'm going to go ahead and get started and give you a quick overview and introduction to why we're concerned about this. Firstly, as I'm sure most of you are aware, low doses of antibiotics have been used as growth promoters in livestock for several decades now. As a result of this, there's been accumulation of these antibiotics in poultry feathers. This is a concern because the poultry feathers are commonly used as a high protein source in animal feed, such as poultry meal and feather meal. The interest here is that the low-dose antibiotics have been linked to the production of antibiotic-resistant microorganisms, which can impact both livestock health and potentially human health. Another area that we were examining in this method is pesticides that can be found in these feeds due to residues in the crops. So when these feeds are produced, obviously there's a lot of grains that are going to these as well, and so we can find these residues. The matrix we're looking at, and this is really a lot of the issue I want to focus around today, is how we work with these complex feeds. They're difficult because of a number of reasons. They have a lot of components there. Obviously, as a feed, they have a lot of fats, proteins, and carbohydrates. But in addition, there's a number of things such as antimicrobials, emulsifiers. We have pH agents, preservatives, binders, etc., which are added to the feed to increase shelf life and to ease production. So it's a very complex system to work with. The analytes that we have in this method is a total of 13. We have 9 antibiotics, which include fluoroquinolones, quinolones, sulfonamides, amphenocols, and macrolides. The 4 insecticides that we are working with are diflubenzuron, emamectin, abamectin, and ivermectin. So the challenge for us as an analytical lab is how do we develop these multi-drug, multi-class methods that can be easily performed in the lab on complex matrices, give you low detection limits, they're reproducible, they're robust, and that can really be difficult in some of these types of matrices. So I'm going to turn this over to Lauren for a second for an audience poll.
Okay, so to get started, we wanted to poll you guys and ask you, what is your biggest obstacle that you have to overcome when developing new residue test methods by LC-MS? Is it optimizing the sample preparation, optimizing the chromatography conditions, optimizing the mass spec conditions, achieving the proper sensitivity for your compounds of interest, overcoming matrix interferences? You face all of those obstacles, you don't face any of those obstacles, or you're not doing any LC-MS method development at this time. So what are your biggest obstacles to overcome when developing new residue test methods by LC-MS? So I'll give just another second or two to get some more answers to come in. And it looks as though, from the current results, we see a lot of people, a little bit of a mixed review right now. More people are showing that overcoming matrix interferences and also optimizing the sample preparation is probably a big part of the obstacles that need to be overcome. So I think, Kurt, this will be really good for your presentation, as you'll be speaking to this. Am I right?
Correct, yeah. We're going to be, that's really the focus of this first part of the talk today.
Okay, perfect. Thanks to the audience for submitting your answers, and we'll let Kurt continue with his presentation.
Okay, so to begin this little next portion here, I'd like to just go over kind of our typical workflow for method development processes at Eurofins. When we go to develop a new method, there's usually kind of 5 steps that we follow through to bring a method online. The first step is taking individual analytes and preparing them as solvent standards, and then optimizing the MSMS conditions, clustering potential, cell exit potentials, collision energies, to really kind of tweak the mass spec to get the max sensitivity. As a next step, we perform flow injection analysis, and this is to optimize our source conditions. We use the chromatography solvents that we're going to be using in the method, if that's known, and it allows us to optimize things such as the electrospray potential, the source temperatures, the gas flows, and so we really kind of get our maximum sensitivity there. Our next step then is to go ahead and develop and optimize the chromatography, and that's pretty self-explanatory. We try to get these first 3 steps in place first before we do a preparative method, so that we know that we have a good analysis technique on hand. Our final kind of 4th and 5th steps here are the actual optimization of the sample prep, and that can be looking at how do we optimize the extraction, whether through differing solvents, differing pH effects, do we do shaking, sonication, etc. We'll also, for a lot of our methods, they'll have a cleanup, whether that's SPE or catchers. It sort of varies in how we take that next step, but we generally have a cleanup step for most of our methods. And then we'll also look at other parameters such as stability of our analytes, stability of our calibration standards, and how long can they sit while they're still good. So that's all important information that we need for these methods. And then our last step is to go ahead and perform a validation of the method, in which we'll essentially turn it over to our lab personnel, have them perform the method, and we'll look at the data and make sure that it meets our performance criteria. And if so, then generally we go ahead and bring that method online. So I'm going to kind of quickly cover a little bit of background on optimizing instrument methods and the chromatography. Saeed is going to talk in much more detail about the chromatography, so I'm going to leave that to him. As I said, when we start these methods, we go ahead and begin optimizing our MS conditions with solvent standards. The instrumentation that we were using for this particular study was an Exogen Expert Ultra LC from AB-SciX. And then also we had an AB-SciX 5500 Q-trap that we're using with electrospray ionization. This instrument was chosen due to basically the rapid speed and sensitivity. The electronics on this allow some really interesting analysis methods due to the speed of the data capture. For this method, we had hoped to get a single run for all of our analytes. We were going to do the quantitation by scheduled multiple reaction monitoring, the SMRM method, which is a typical way to quantify analytes by MSMS on AB-SciX instruments. And we used polarity switching to collect both positive and negative ion data in a single run. So this allowed us to do basically take 2 separate runs, combine them down to 1, and really increase our throughput. Like I said, on the chromatography, I'll briefly cover this, and Saeed will go into more detail. We worked with Phenomenex, and they developed a rather unique column configuration for this analysis that uses a pair of security guard cartridges ahead of a silica mixer column that sits before the actual analytical column, which is the Gemini NXC18. This was used to overcome some issues with early eluting peaks and bad peak shape. And you'll see some of the SIEDS data, and it's a phenomenal difference using this type of column configuration. The mobile phases that we had were water with 0.1% formic acid as mobile phase A, and then 10 millimolar ammonium formated methanol as our mobile phase B, and then a fairly standard gradient. This gives a little bit of a taste of kind of what we have for the chromatography and how this looks. This is simultaneous data collection of positive and negative mode ions. The 5500 allows a 2 millisecond switching time between polarities, so that we can simultaneously collect these. And this is, I believe, a 40 ppb solvent standard. And we have really excellent peak shape, and it works quite well. Just for your information, these are the MRM conditions looking at the transitions for our quantitative ion and our qualitative ion, qualifier, and along with the retention times. And you can see in here we have two ions that are collected in negative mode, which is the 4-phenol and the chloramphenicol. So now I want to get into kind of the meat of what I'm going to be talking about today, which is the method development process of the sample preparation. And that includes the extraction, the cleanup, and reconstitution and dilution of the sample at the end. We looked at a number of factors in developing this method. We optimized our extraction solvents in terms of stability, looking at pH effects on our analytes. We looked at absolute recovery, obviously that's important, as well as compatibility with our SPE cleanup method. We also examined the impact of the extraction solvent on the instrument response in terms of peak area as well as signal to noise. And then we also looked as well at the extraction solvents in terms of sample stability in the different solvents that we were looking. We have a number of slides examining SPE elution efficiency and some breakthrough studies that were performed to look at our solvent composition. And then lastly, we did some work looking at the effect of drying and reconstituting samples as far as, what were the temperature effects, how far could we dry down, what were dilution factors at the end, and how that impacted the method. So the first little set I'm going to talk about is the analyte response to signal to noise and potential elution solvents. The SPE cartridges we're using are the Phenomenex Strata XL-CW, and these require an acidic elution solvent to release the analytes. So we knew that we would have to look at the impact of pH on our analytes on the instrument. So we evaluated in terms of peak area and signal to noise. And the following elution solvents are what we looked at: 0.51 and 5% formic acid in methanol, and 1, 5, and 10 millimolar oxalic acid. That was also in methanol. It's not listed on the table, but that was in methanol. As you can see, the pH range on this range is from 2.85 to 1.98. So these are pretty acidic. So the first thing we investigated is taking our combination of our analytes and a solvent standard, and we compared that against our different potential elution solvents here. And as you can see, if you look on the left, our y-axis there is actually the log of the peak area response. So as you look between the formic acid samples and the ones that were eluted, or potentially eluted in the oxalic acid, we see a much higher signal response from the oxalic acid by about an order of magnitude. And that pretty much carries across the board for these samples. So there's obviously an impact of the oxalic acid. However, when we looked at the signal to noise, which is really what we're more concerned with than anything, because the signal to noise determines where we can set our LOQs and our LODs, we don't see a huge difference. There is some increase in signal to noise in oxalic acid for a few analytes, but overall, we see pretty similar responses. And sort of what we're looking for here is we want the average signal to noise to be, pretty good. So if we have, some that are really poor and some that are really bad, or some that are really good, that doesn't do us a lot of good for multi-class methods. So we try to get, sort of a compromise of the signal to noise values. And to that regard, the formic acid really did perform a little bit better than the oxalic acid. Next, we looked at the analyte stability if we were to take these and hold them in these solvents for a period of time. and dry them down. So we prepared our standards in these various solvents, different concentrations of oxalic acid and formic acid in methanol. We dried them to different degrees, either completely to dryness, down to 1 mil. And then we also looked at the impact of buffering the oxalic and formic acid before drying. Our concern with the pH effects were, as we take a fairly acidic solvent and we begin to dry it, we were driving down the pH by driving off our solvent there. So we were concerned that we were going to hit a point where the pH effects would become really kind of severe and damage our analytes and degrade them. And we found that with the buffering, there is some impact. We do find improved recoveries relative to the non-buffered systems. So in the next section here, I'm going to cover a little bit of the work we did looking at some breakthrough studies and the effects of dilution of our final eluents off the SPE cartridges. So in this is sort of a rough breakdown of what we did for the method. We had 5 grams of sample that was extracted with trichloroacetic acid and EDTA in sequence. And then we added methanol and water and combined these extracts. They were brought to a total of 50 mils. And our final methanol concentration in these is about 10%. And that becomes relevant in the next few slides. I'll discuss that. We then conditioned our SPE cartridges, put our analytes through, and then we eluted them with either 10 millimolar oxalic acid in methanol or 5% formic acid in methanol. We took this final eluent and we diluted these down, either non-diluted, the 1x solution, or 2 or 4-fold prior to analysis. And what we're looking at here is we were wanting to see the impact of dilution upon matrix effects and seeing if that affected our signal to noise levels. So it's kind of a little breakdown of what we found from this study, and there's a couple more slides with some data on it. We found that the elution in the oxalic acid in methanol and the formic acid in methanol gave equivalent recoveries. There really wasn't much difference there. We did find, though, that our peak shape using the formic acid was somewhat better than oxalic acid. So there was an impact on the chromatography, and so we kind of decided in our final method, as you'll see towards the end, that we ended up going with formic acid, and this is one of the deciding factors. Another interesting thing that we found here is that we had some significant signal-to-noise improvements using larger dilution factors. Even though our absolute peak area was lower, our noise dropped out faster, and so we were, in essence, due to the sensitivity of the 5500, able to basically reduce our limits of detection, limits of quantitation by using a larger dilution factor at the end. And we were able to recover all of our analytes with the exception of tetracycline at the 1x dilution, and we suspect that might be due to pH effects, and that diluting those down helped with that. So this gives a little description of the signal-to-noise dependence of the dilution factor on a number of analytes. So we see here on each of these little plots is the left-hand bar is the 2-fold dilution, and the right-hand is non-diluted. And generally speaking, we see anywhere from a 1/3 to double of our signal-to-noise increase, which is pretty significant. And so that tells us that we're able to kind of improve our detection limits by diluting this out a little further. The next thing that we looked at is some breakthrough studies looking at the dependence of the methanol concentration and seeing basically how high of a methanol concentration can we use in the extraction before it affects our SPE cleanup. Obviously, for these nonpolar organic compounds we're looking at, we want to kind of maximize our organic extraction solvent content. So we loaded matrix match standards onto the SPE, and then we eluted 3 times with the elution solvent, which contained either 0, 5, 10, 25, 50, or 80% methanol. And what we found was that for all fractions that were collected with less than 10% methanol, we saw no breakthrough in the first 5 mils of the collection, which is kind of what we were hoping for as our final elution volume. As we increased that to 25 and 50 and eventually 80, you can see that we pretty much wiped everything off of the SPE by using that. And so in that first 5 mils, it tells us that we really are limited in the absolute amount of methanol we can use on the SPE. And the general consensus is that we had to use less than 25% methanol in our extraction solvent for the method. The next step that we looked at is looking at the effect of the bed volume of the SPE cartridge as well as the sample loading by changing the sample mass and changing the final volume to basically normalize everything. And what we found is that there really isn't a whole lot of difference on whether we use the 200 or the 500 milligram cartridge. Overall, the 200 milligram using a 2.5 gram sample loading gave us the best average recoveries, but if you look at these numbers in detail, there isn't a great deal of difference. So what we decided from this is that using the lower bed volume would be optimal because we can obviously, we can elute faster through these with the smaller volume. And so it helps us kind of speed up the method and use less solvent. And so that was kind of where we moved forward from there. So at this point now, we have essentially a finalized method based on all the data that we've compiled. As you can see here, what we concluded is using a 1.25 gram sample, and this is wetted with water first. And for anyone that's worked with dry feeds before, that wetting step is we find it to be quite important actually. That if you move straight to an extraction without the wetting step, your recoveries are much lower. We then added 5 mils of 1% trichloroacetic acid, followed by 5 mils of 1.5 millimolar EDTA, and those are just vortex shake, centrifuge, and decant. As our next step, we added 10 mils of 75% methanol as an extraction step. Now, when we add the 75 mils, or the 10 mils of 75% methanol, our final volume after combining all of our extraction steps is 50 mils. So if you run through the numbers on there, that works out to a 15% methanol concentration in our final combined extract, which was below our required criteria of 25% to make sure that we don't have breakthrough of analytes. So our final step before going to the SPE then was we would add 10 mils to the sample and 10 mils of water to the sample and go through the same process, vortex shaking. We'd combine the extracts and then bring those to 50 mils of water. We then run through the SPE cartridge, which is a 200 milligram, the Strata XL-CW. We would wash with 10 mils of 15% methanol to ensure that we keep our analytes on the column. And then finally, we would elute with 5 mils of 5% formic acid and methanol, evaporate the samples down to dryness, and reconstitute in 70% methanol, filter, and they're ready for analysis. So gives a little idea of the analyte recoveries of our finalized method. We have our 13 analytes on our left, as well as our linearity, the R squared of our calibration curve, and these are matrix match standards here. And you can see that our linearity of our cal curve is excellent, 0.997 or higher, well actually 996 or higher. As far as our analyte recoveries, we found that all but 4 of the analytes recovered about 50%. And we did not use deuterated standards when developing this, but if we were used to use deuterated internal standards, these recoveries could be improved significantly. And I suspect that we would be looking more in the area of, hopefully everything above 50 certainly. But considering the sort of the wide variety of the chemistries of these individual analytes, this is, I think, is pretty good, and we were happy with this. Lastly, I'll kind of just go through the conclusions of what we found from the study, is that we were able to develop a method which could rapidly analyze a mixture, multi-class, multi-residue mixture of antibiotics and insecticides. We had a unique chromatography column design, thanks to the people at Phenomenex, using a mixer column, analytical column in line. And by using the 5500 Q trap, we were able to do simultaneous collection of positive and negative ions. And sort of lastly, we went through and showed the steps here where we were able to optimize this preparatory method using a variety of factors from the extraction, cleanup, reconstitution, and dilution. Quick look at references, and lastly, I would like to thank our collaborators, Lauren Bailey and Andres Schreiber at ABSciX, at Phenomenex, Saeed, Marco, I'm not going to attempt to pronounce Marco's last name, and Sky Countryman. And then here at Eurofins, we had a number of people that worked on this project. Ryan Piku did the bulk of this work, Sarah Ruiz, Cheryl Stevenson, and John Ruther. And that concludes my portion of the presentation. I'll turn it over to Lauren and Saeed.
Thanks, Kurt. So I'm going to turn it directly over to Saeed, who will continue with discussion of the LCMS method development and the chromatography. And just to remind everyone, if you do have questions for Kurt or Saeed, you can put those questions in the top left-hand box on your screen. Saeed, take it away.
Good morning. Thank you, Lauren, and thank you, Kurt. Initially, when we were involved in this project, the scope of our analysis was to figure out how to get these compounds to be analyzed under one roof in one setup. As Kurt mentioned, they all have different chemistries and they don't behave ideally under the similar conditions. So the scope of analysis were basically three parts: to look at the chromatography setup, or more accurately LCMS setup, and we found a few interesting tidbits about these compounds, which we will point out, along with finding out some issues in the ionization source, and finally we'll wrap things up with the sample diluent. So the group that kind of attracted our attention were the macrolides, specifically the mectins. They seem to be a little bit different. There was not much of a knowledge about their behavior. The other compounds, there was adequate literature, and the chemistry had been studied fairly extensively. I'm going to let Lauren take a poll here, and take it away, Lauren.
Okay, so we'll take one quick break before Saeed continues and do another polling question. So now we want to ask, how many compounds or analytes do you typically screen for or quantify in a single analysis? Is it between 1 and 5, between 6 and 20, between 21 and 50, between 51 and 100, or more than 100 in a single analysis? So how many compounds or analytes are you typically screening or quantifying for in a single analysis? And we'll go ahead and broadcast the results, and what we can see is most of the results are showing a mix between anywhere between 1 compound and 50 compounds. So Saeed, keep that in mind as you're going through your talk about developing your multi-residue chromatography method. So I'll hand it back over to you.
Okay, thank you. I should commend those people who look at more than 50 compounds. It is not an easy process. So let's look at these mectins. This is what they look like. They're fairly similar. There are some differences between these macrolides. The R group in the corner, upper right-hand corner, it's either CH3 or C2H5, a methyl or ethyl group. And even though they do look a lot alike, they don't quite behave even the same way. So the MS method that we developed, it was fairly standard. We looked at some of these compounds. We've seen, all right, most of them form a protonated ion in the source. A few that was mentioned earlier, they come up with a deprotonated form as the prominent ion, and a couple of these mectins seem to require ammonium buffer to form an ammonia adduct. So unfortunately, we will also see sodium everywhere in some of these compounds. So as we began the development, we discovered some physical property with the mectins that kind of threw a monkey wrench in the process. They're very hydrophobic. they require high organic solvent to be stable. We made a 5%. We made a solution in 5% methanol, and we have about 1 injections out of that solution before we lost the mectins. Unfortunately, well, if you have high organic concentration in a sample, that would constitute a strong solvent on a reverse phase chromatography, and the consequence of that is what you will see is a poor peak shape and analyte breakthrough as this analyte is injected in a plug of high concentration organic. It goes from auto sample pretty much intact, lands on the column, and the organic solvent goes through the column and drags the analyte with it. So regrettably, we have to do something here to fix the situation. The solution we came up with was, well, why don't we dilute the sample while it is traveling from auto sample and onto the analytical column? The choice or the solution was insert a small silica column, in this case Luna 5 micron silica post-injector valve. As the plug of sample goes through this cartridge or this column, it will begin to diffuse, and the organic solvent mixes with the mobile phase. By the time we reach, or the analytes reach the analytical column, it will be in a mixture that is fairly close to what the mobile phase conditions are. So in this case, the blue arrow indicates where the sample comes. It goes through that silica column and onto the Gemini NX analytical column, and then into the mass spec. So this is what happens when we put that silica column. The peak shape improved nicely. The breakthrough is gone. To give a better perspective, I'm going to put them together. So the peak or the trace at the top is with the silica column, and the trace at the bottom is without that mixer column. One thing I should point out is the peaks elute a little bit later, about 20 to 25 seconds later, and that's because there is an added system volume in the chromatographic system, so that delays the chromatography a little bit. And the next question that we came across was, is it possible that we could inject too large a volume and lose the mixing ability? So in this case, we picked a fairly viscous solvent, isopropanol, as our mixing solvent. We placed the sample in 100% IPA, made several injections. The limitation in the injection volume is generally seen by the first eluding peaks, because they are not retained very well. So as you can see from the top trace, 15 microliter, the peak looks really nice. And the bottom trace, 25 microliter, you're starting to see some form of a peak fronting. So at this point, our conclusions was probably 25 microliter would be the maximum we can inject on a fairly viscous solvent. The final LC method we developed was using a 2 sets of column Gemini NX C18 50 by 2 3 micron particle size, and a Luna Silica 50 by 2 with 2 guard cartridges, or a 30 by 3 millimeter 5 micron. They both have similar volumes. And formic acid and methanol with some ammonium formate in the methanol to enhance those mectins that I mentioned. Obviously, this needs to be modified from LC system to LC system. I believe at Eurofins, the LC system that was used was different than our adjuvant. So we fixed the chromatography to a point where sample can be placed in a suitable solvent to resolve the hydrophobicity issues of some of the analytes. As we found out later, mectins are also fairly susceptible to thermodegradation, and they're also susceptible to photodegradation. In this case, what was what became an issue was the temperature of the drying gas. That's gas 2. If we reduce the temperature, we can get a little better stability of ions in the source before they're analyzed. So we have two sets of temperature. We followed pretty much the same path as Kate mentioned to optimize the source conditions, specifically the gas temperature, the drying gas temperature. But afterward, we found that mectins require different set of conditions. So in this case, if you take a look at the trace, the top trace, amomectin is fairly stable, but abamectin and ivermectin pretty much disappear at 500 degrees Celsius temperature. But at 400 degrees Celsius, we do manage to see both abamectin and ivermectin, even though we do lose some sensitivity for amomectin. I'm going to pull out the two smaller signals for ivermectin and abamectin so we can have a little better look at the sensitivity of these two at different temperatures. As you can see, ivermectin has a far better signal, as well as abamectin at 400 degrees Celsius drying temperature. I believe the not full order of magnitude, but close enough so at least they will be stable. So then the next obvious question was, all right, we can see the abamectin and ivermectin that elute at fairly high organic concentration in the mobile phase. What about the early eluting peaks that don't have fairly good signal and they elute at mostly aqueous components? So the mobile phase is harder to desolvate. Do we lose some signal? The answer is yes. Is it catastrophic? No. So at 400 degrees Celsius, we can still have good sensitivity, even though we lose a little bit of the signal. So our conclusion was, yes, we're going to have to make some kind of a compromise, but at least we can see all the analytes. And these are the conditions we ended up using on our API 5000. Gas 1 and gas 2 at 60 and 50, and temperature for gas 2 at 400 degrees Celsius. So if you've followed us to this point, our effort has been to figure out some form of a stable situation for these analytes so they can be analyzed while waiting in the other sample vial. The next question that came up with, in addition, can we do something to enhance the sensitivity while we have these analytes, or put something in the solvent to increase their sensitivity? We picked the tetracyclines in this case. As Kit mentioned, it requires, it seems, to have a more acidic environment for it to have better sensitivity. If you take a look at this compound, it has multitudes of functional groups that can act as chelating agent, host either an acidic proton, sodium, or stick to silica, make chromatography a little bit more difficult, and also a number of functional group, 2 enols and a phenol group that can act as acid. Those protons can leave and form a negatively charged species that would counteract the charge that might be on the other side of the molecule, hence losing sensitivity because of this behavior. So we took the 4 tetracyclines, we tested them at 1 ppb, 1 nanogram per mil. In this case, we picked the Luna column. We were testing tetracycline, oxytetracycline, chlorotetracycline, and doxycycline. So that's a little fast to say all these. So the first compound of interest was tetracycline, and the last one was doxycycline. We made 5 different solutions, different acid strength, and different pH. In this case, we're looking at the chromatography to make sure there's nothing that these acidic solutions will do to the peak shape. I have overlaid all of these peaks, and as you can see, there's nothing that's that make them any different, any unusual peak shape or difference in the retention time. On the next slide, what I've done is I've used analyst features and moved the peaks around, made them offset so we can see their sensitivity. So as you can see, the first group are tetracyclines and tetracycline itself. The second one is doxycycline. Our first baseline was the samples were prepared in 0.1% formic acid, and it has about a peak height of about 9,000 counts. As we increased the concentration of formic acid, we didn't really gain much. Maybe we took a little bit of a hit on the sensitivity. But as we reduced the pH, in this case we used 0.01% TFA, and then phosphoric acid, the signal is increasing. Nobody would obviously put these in the system. That is, I've never heard of anybody use phosphoric acid as a mobile phase in an LCMS solution. And finally, 10 millimolar oxalic acid, which has probably the lowest pH of all of these. And what you can see is the early eluting peak shows fairly good enhanced sensitivity as pH drops, even though we're using a conventional mobile phase for our chromatography. We're just changing what's in the sample. Even the late eluting peak, the doxycycline at 2.5 minutes, it is showing pretty much the same trend as pH is reduced. Even though 2.5 minutes later, we are still getting good signal for these compounds. So our conclusions are, obviously, we've had to figure out a way to make all these compounds to stay in solution long enough to be analyzed by the mass spec. So we did make some compromises, and we came up with a solution for all to be stable. And further, it seems we had to make more compromise in case of the mass spec source to make sure the compounds don't disintegrate in the ionization source. And finally, addition of that mixer column post-injector enhances the chromatography, allows us to go and pretty much have an easier hand at making the sample solvent more agreeable to everything. In final, I'd like to thank everybody at Eurofins, Cheryl Stevenson, Ryan Piku, Davey Sykes, Lauren Bailey, and Team Phenomenex, specifically Mr. David Trine. So thank you very much. That's it. I'm going to turn it back to Lauren.
Thanks, Syed. So we'll start the question and answer portion. If you have questions, you can submit those in the top left-hand box of the screen. And I'll start with the first question, which is probably for Kurt. And the question is, have you been able to apply the sample prep approach that you developed to other matrices other than feed?
This specific prep, we've used this in a number of different types of feeds. It's sort of a similar extraction, this multi-step extraction procedure. But no, we haven't looked at other matrices such as tissue or things like that.
Okay. The next question I think probably also will be for Kurt, and the question is, why do positive and negative analysis at the same time?
It's essentially to speed up sample throughput. If we have 2 chromatography runs that are running at 10 minutes each, if we can do simultaneous analysis, it cuts our analysis time in half. And we're a very high throughput lab, so anything we can do to reduce our analysis time is a big help to us.
Great. The next question is for Syed, and the question is, the run appeared to be 13 minutes long, and have you considered any chromatography to shorten that injection time?
The answer is yes. During the initial setup, when we were running the chromatography, we wanted to make sure both columns were in stable condition before the next injection. So we took a little liberty having a longer equilibration time. But if you notice, we use a 2 millimeter ID column. The optimal flow rate for that particular ID column is 0.4 to 0.5 mil per minute. We ran the mobile phase at 0.7 mil per minute. So it was probably more of a choice on our part to be cautious than it was required probably. But yes, probably 10 minutes would be adequate for this.
Okay, great. Thank you. Thanks to Kurt and to Syed for their presentations today. And thanks to everyone here for attending the webinar. We just want to ask if you can take 1 minute to tell us what you thought about the presentation. If you can tell us how you would rate the webinar on a scale of 1 to 5. And also, if you have other topics of interest that you would like to hear in future webinars, go ahead and type those in as well, and we can consider that for future webcasts coming up.